So we are continuing our discussion of molecular basis of inheritance. So this chapter is present in your class 12th NCERT textbook, and the topic of discussion of this particular video would be Hershey and Chase experiment. Now, very important to understand what happened before this. So, if you see my previous videos, you will understand that initially the scientists were confused that which biomolecule will actually store the genetic information, whether it will be DNA or protein. And we knew that something came unexpectedly from an unlikely source. That was an experiment by Griffith, where he discovered the process of bacterial transformation. And then we needed to find the transforming principle, and if we are able to isolate the transforming principle that transformed a type of bacterium in Griffith experiment into another type of bacterium, then we will get the biomolecule that will be storing the genetic information. And I told you in another video that this thing was initially done by Avery, MacLeod and McCarthy. So they worked with the lysate of the heat killed S, and then they removed the various biomolecules and reached the conclusion correctly that it will be the DNA that is the genetic informational molecule, or the molecule that stores the genetic information, and not the proteins. But there was a line of NCERT. There was a line of NCERT that I pointed out in the previous video. Your NCERT said that not all biologists were convinced. So we know that Avery, MacLeod and McCarthy were able to isolate DNA in pure form, and this DNA was able to bring about the transformation of the bacterium. And if we digested DNA by DNAs, then transformation was not possible. That was a direct evidence that it has to be DNA. Yet my NCERT says that not all biologists were convinced, and I told you the reason about it. I told you that at that particular time, the model that we were aware of the structure of DNA was actually given by Phoebus Levine, and it was a tetranucleotide structure with fixed repetitive sequences, and the scientist at that particular time actually believed that DNA molecule will not exhibit diversity. And since we need a particular biomolecule that must be able to explain the diversity of the living world, that is why not all scientists were satisfied with the result of Avery, MacLeod and McCarthy. Now, that is why your NCERT says that this unequivocal proof was given by Hershey and Chase. So why did we require this unequivocal proof? Because not all biologists were convinced because of Avery, MacLeod and McCarthy, and therefore we needed an unequivocal proof that what Avery, MacLeod and McCarthy are saying is true, and it is DNA and not protein that will store the genetic information. So a very elegant experiment was performed by Hershey and Chase to prove this. Now please understand a very important point right at the beginning. The success of any experiment will primarily depend on the material that you have chosen as the experimental material. So suppose I want to say that cigarette smoking causes cancer. So at least I will choose the people who actually smoke cigarette. Otherwise it carries no meaning. So that means whenever you perform an experiment, you will have to choose very judiciously and meticulously the material that you will like to work upon. And this is very important. This is exactly what Hershey and Chase did. So they worked with what is called as a bacteriophage. So we know that bacteriophage are the viruses that infect bacteria, and we know that viruses are nucleocapsid. That means the two components of virion or virus particle, one is a protein coat that is called as capsid, and there is a core which contains the genetic material. And this bacteriophage, their genetic material is double-stranded DNA. So that means we are looking at a particle that has double-stranded DNA as the genetic material in what is called as the core region of its particular structure. It is surrounded by a protein coat, and this protein coat is called as the capsid, and I am calling virus as a nucleocapsid. I hope the terms are absolutely clear. Now please understand, what we are saying that we are confused between DNA and proteins. And can you see The choice of bacteriophage is critical here because it is made up of only these two things that we are confused in. So it is made up of only DNA and protein. So that means we will be able to reach the correct conclusion by excluding either of the two. So that means this choice of the material was very critical. Again, I am telling you, because we are confused between two biomolecules. One is DNA and other is the protein, and that is why we have chosen a very important particle which is actually made up of these two things only, which is double-stranded DNA and protein. And again I will repeat myself. I have already told you this thing in previous videos, that most of the experiments which involve bacteria in molecular biology have been done on E. coli. So that means this is the bacteria which is Escherichia coli, which is involved in majority of the experiments. Since we have read most about E. coli and we are absolutely sure about so many aspects of the E. coli because we have read it very well, so we are most comfortable working with E. coli. This is the reason why in molecular basis of inheritance most of the times you use this bacterium E. coli. So suppose the examiner asks why E. coli, because we are most comfortable with it. Why we are most comfortable with it? Because we have studied this bacterium in detail so many times and we know almost all the genetics of this particular bacterium. So now there are two things. One is a bacteriophage. This bacteriophage will be infecting this bacterium. So we are taking a bacteriophage with double-stranded DNA as the genetic material, and the bacterium is E. coli. So now let us see what they did. So two things were very important in this particular experiment. One, the choice of the material, as I told you. Second, if we look at DNA molecule, then we say that DNA molecule will contain phosphorus. So DNA molecule has phosphorus contained within its structure. You know how nucleotides are made up of. So nucleotides are made up of a pentose sugar, a nitrogenous base, and phosphate group. So that phosphate contains phosphorus, but it does not contain sulfur. So that means in DNA you have phosphorus present but not sulfur. But on the other hand, the other biomolecule of contention, which is protein, it is the reverse. It is the other way around. That means it will normally not contain phosphorus, but it will contain sulfur. So the idea was to differentially radioactively tag the two. So you can tag DNA radioactively by using a radioactive isotope of phosphorus, and you can tag the protein by using a radioactive isotope of sulfur. Sulfur radioactive isotope, if you are doing for tagging, it will not tag DNA, and if you are using a radioactive isotope of phosphorus, it will not tag the protein part of it. I hope this is absolutely clear. So now we have bacteriophage. It is made up of only those two particles. Again, I am telling you that we are confused. That means it is made up of only double-stranded DNA and protein. And number two, we can differently radioactively tag them differentially. So we can either tag phosphorus or tag sulfur, and we know that DNA will carry phosphorus but not sulfur, and protein will carry phosphorus. Proteins will carry not the phosphorus but sulfur. You know there are sulfur containing amino acids. There are two sulfur containing amino acids. Please make a note of it. One is cysteine and the other is called as methionine. So these amino acids will be incorporated into the primary structure of the protein, and hence we know that protein contains sulfur. I hope this is absolutely clear. Now what they did, they actually grew a colony of the bacteriophage in an environment that was providing the radioactive isotope of sulfur. That means they grew them in 35S. This was the radioactive isotope used by them. As expected, this will tag the protein and not the nucleic acid. So which means that if we look something like this, that they were able to tag what is called as the protein part, but the DNA was not radioactively tagged. But if they are using 32P, this is the radioactive isotope of phosphorus, we know that it will tag the nucleic acid and not the protein coat. So in this case, these particular phages, they have normal or not radioactively tagged protein coat, but their DNA will be radioactively tagged. So this was very easy. So you have generated a colony of bacteriophages there where you have radioactively tagged the protein part by using this appropriate isotope, which is 35S, and you have tagged DNA by using 32P. This was a big advantage of using bacteriophage again. This is what I told you in the beginning as well. So now we have some bacteriophages with radioactively tagged protein but normal nucleic acid, the others which have radioactively tagged nucleic acid but not the proteins. So what is the next step then? So the next step was that they separately allowed these bacteriophages to infect the E. coli bacterium. So let us take a test tube. And we are saying that in this test tube the bacteria are allowed to be infected by the bacteriophage, and these bacteriophages are having 32P, which means their nucleic acid is radioactively tagged but protein is not. And similarly here we are saying we are using the bacteriophages which are tagged by 35S, and we are allowing a separate colony of E. coli bacterium to be infected by this particular phage. In this phage we know that the protein part will be radioactively tagged but DNA will not be tagged. And as we are aware from our previous classes that what happens when a bacteriophage infects the bacterium is the fact that this bacteriophage will first attach to the receptors which are present on the bacterial cell. So that means the phage is not going to enter into the bacterium. It will remain on the outside and it will bind to the receptor that are present on the surface of the bacterium. This will provide specificity. Therefore, you know that a particular type of phage will be having a host range. It will be able to infect only some bacteria, not other. And this host range will include the bacteria that carry the receptor on the surface for this bacteriophage. This is very, very important to understand. So we have taken a phage that is capable of infecting E. coli. That means on the surface of the E. coli are present the receptors for this bacteriophage, and the bacteriophage binds to the receptors. Then you know from your class 11th there are this tail part undergoes contraction and it pushes the double-stranded DNA into the bacterium. So it pushes the double-stranded DNA into the bacterium, whereas the capsid part will remain on the outside. This is what we know. You have read in your class 11th NCRT the lytic life cycle, the lysogenic type of life cycle in the viruses. So this is what we are saying, that when a bacteriophage infects a bacterium, it injects or transmits the DNA into it. You can see that this particular thing actually, which is the capsid, remains on the outside. It is very important to understand that it does not enter the bacterium. Now what happens? This double-stranded DNA will take control of what is called as the main chromosomal DNA of bacteria. We know that some bacteria may have extra chromosomal circular double-stranded DNA called as plasmid as well. But what I am saying that this is the main chromosomal DNA. You know, bacteria normally has a single chromosomal DNA which is circular and double-stranded. So this double-stranded DNA molecule of the bacteriophage is going to take control of the main chromosomal DNA of the bacterium. So now the metabolic machinery of this particular cell is under the guidance of this DNA, which actually belongs to the bacteriophage. Now what is going to happen then? Then what is going to happen? There will be replication of these DNA particles. So these DNA particles will be replicated. Their genes will be transcribed. You can understand on this double-stranded DNA there will be some genes located, and these genes will be transcribed and translated into proteins, and these are the proteins that are the part of the capsid of the virion. So please understand, that means on this DNA we expect to find some genes that will make up the proteins that make up the capsid. So that means the genes are transcribed and then translated, and then with the help of these proteins there are formation of capsids inside the bacterial cells. So that means there are new capsids which are being formed. Now the last step is that whichever genetic material, whichever double-stranded DNA has a particular sequence of nucleotides which is called as the packaging sequence, will get incorporated into the capsid. So please understand, only those double-stranded DNA molecules that have packaging sequences will be packaged into the phage particle, and this new particle will be called as virion. It may decide to kill the cell or they may leave the cell as well, depending on whether it is a lytic life cycle or a lysogenic life cycle. So we know this. So Hershey and Chase were also aware of this particular thing, that how a bacteriophage infects a cell or a bacterial cell. I hope this thing is absolutely clear. One more point to note here is that once the infection has occurred, which means the phage has been able to transfer the double-stranded DNA into the bacterium cell, this capsid will become empty. Now it does not contain DNA. It will look empty. You can easily understand. It contained initially the double-stranded DNA, but the double-stranded DNA has been passed into the bacterial cell, and if this infection has occurred, we know that this will appear empty. And if we look at this particular phage at this particular time with some special techniques that you will learn in higher classes, this will appear as what is called as a ghost. Now ghost means that it is empty and it will look empty, and therefore they started calling them as ghost. And once you are able to see the ghost that is devoid of the nucleic acid, you know that the infection has occurred and the double-stranded DNA has been passed into the E. coli. So now please understand what is happening here. So in this test tube which we are labeling as A, and in this test tube which we are labeling as B, here we have added the bacteriophages with radioactively tagged DNA, and here we have added bacteriophages with radioactively tagged protein, and we are allowing the infection to occur, which means we are looking for these ghosts. So what is going to happen? The bacteriophage will join the surface, will bind to the surface of the bacterium, and then will transfer their double-stranded DNA molecules into the bacterium in both the cases. And you will instantly come to know that infection has actually occurred when you see what are called as the ghost. So let me make something like this. So what we are saying, we are looking at bacteriophages which are now seemingly empty, which means now we are convinced that the bacterial cells have been infected. So this is a very, very important point to understand. Now we know that infection has already occurred and we are able to see the ghost. So this is what they allowed to happen. That means they allowed the phages to infect the bacterial cell, and therefore we know that the double-stranded DNA has been passed into the bacterial cell. So this is very, very simple. This is very, very simple. Now what they did, they used a blender. So blender is something what is called as a mixie in your kitchens. So this was a very simple experiment, and most of the experiments in molecular biology, you will see they are very simple, and simple means elegant. That means if something is very simply explained, then it looks very elegant, and that is the purpose of the science. Science tries to follow the principle of parsimony. It tries to simplify the things that you are aware of. It tries to tell you something in a language that is very easily understood, and it should not be too much complicated as well. So you can see there is nothing complicated about this experiment. So that means they took a blender, kept their tubes into the blender and started the blender, which means there will be dissociation of this particular phage from the bacterial cell. So that means now they wanted that once the infection has occurred, they must remove the phage particles which are attached to the bacterial cell. They do not want the association of the protein in any way to the bacterial cell in such a manner that they may actually be influencing the process of the transformation, etc. So that means they wanted to remove the bacteriophage from the bacterial cell, and for that they did what is called as blending, and they use a kitchen blender for that. Can you imagine? And then what happened, that after some time the bacteriophages were removed or were getting dissociated from the surface of the bacterial cell, and then they allowed the contents to stand still so that sedimentation occurs. So what is happening? I have put this tube, I have allowed the infection to occur, I have put the tube into the blender, I want the phage to be removed from the surface of the bacterial cell, and then I allow sedimentation. So after sedimentation, in this case, what is going to happen? There will be something which is collected down and something which is collected upside. This is called as the supernatant. And that means this is very, very easy to understand, because bacteriophages are much smaller than the bacterial cell, and therefore bacterial cells are actually heavier than the bacteriophages. So in the lower part, we expect that what will be getting sedimented, this will contain the bacteria, and the supernatant will contain the phages. So please understand, this is happening now. Now what is happening here? Again there will be a supernatant and something down below, and again this will contain bacteria, and this will contain the phages that have been dissociated by the blending from the surface of the E. coli cell. I hope this thing is absolutely clear to everybody watching this video. Now it was very simple. Now just we have to see that where is the radioactivity. So just we have to see that which part contains radioactivity. And as expected, you know that in this experimental setup we use 32P. This 32P radioactively tagged DNA. This DNA during infection enters into the bacterial cell. Then we separated the phage from the bacterium, and in the supernatant part there was the collection of the phages, and in the lower part there were collection of the bacteria. So we expect that in this particular setup, the radioactivity will be seen in the lower part. So this is absolutely clear. So you will see radioactivity, and you will see the radioactivity within the bacterial cell. So you will see the radioactivity within the bacterial cell, and you will see no radioactivity in the supernatant. The reverse will be true for this experimental setup, because we have used 35S, which radioactively tags the protein and not the DNA, and we know that we have removed the capsid from the bacterial cell. Then you will see that the radioactivity will actually be seen in the supernatant. So when you are using 35S that tags the protein part, almost all the radioactivity will be seen in the supernatant, and no radioactivity will be seen in the bacteria. Now you know that these bacteria will reproduce as well, and you will see the radioactivity in some of the progeny as well. So please understand, very important thing, not only I am seeing radioactivity in this particular part within the bacterium, but when I see that these bacteria reproduce, they produce progeny, the progeny also have radioactivity, at least some of them. This was a very, very important thing. Now you have understood the experiment. So let me just recap the experiment, and then we will see what conclusion can be drawn. So first, I want to prove whether it is DNA or protein. These are the two molecules I am confused in. So I have chosen a material which is a bacteriophage, which contains only these two that I am confused in. That means it is made up of DNA and protein. And I am taking a bacteriophage that is capable of infecting E. coli because I am very comfortable with E. coli. One more advantage of using phage was that since it contains only of DNA and protein, I can differentially radioactively tag them. So I have created a colony of the bacteriophages that will be tagged by 35S, that will tag only the protein part, not the DNA. On the other hand, if I use 32P, it will tag only the DNA and not the protein part. So I have two colonies and I am allowing them separately to infect E. coli in two different test tubes. I know that the infection will be apparent when I am able to see the empty phages or the ghost. So then after the infection, I did the blending part. So that means I removed from the surface the phages. I do not want in any way the protein to be associated with the bacterium. So in the blending part, you dissociate the two things and then you allow this thing to settle down and you allow the sedimentation to happen. So there will be a lower part, there will be a supernatant. And then you saw that when you use 32P, the radioactivity was within the bacterial cell, not only within the bacterial cell but within the progeny as well. And when you are using 35S, none of the radioactivity was seen in the bacterial cell, or almost all radioactivity was limited to what is called as supernatant part of the sediment. And very importantly, these bacteria, when they reproduced, none of the progeny had radioactivity. Now that means the inferences are absolutely straightforward. So what enters into the bacterial cell? Only DNA. Protein does not enter into the bacterial cell in any way at all. Number two, we have removed the protein from the surface. So that means if any signal transduction has been done because of the protein, that possibility has also been removed. It is no way influencing the bacterial cell, and yet the bacterial cell has been transformed. And this transformation, we know, is the permanent genetic change. We are seeing the radioactivity being passed on to the progeny as well. So it is the only DNA that is entering into the bacterial cell because of the radioactivity we are finding inside the bacterial cell if we use 32P. And these bacteria, when they produce progeny, some of the progeny are also radioactively tagged, which means that they have been transformed, and this bacterial transformation is a permanent genetic change. This part is very, very important. So now at this time it was absolutely clear that it has to be DNA. It was a reinforcement of the deductions that were actually achieved by every MacLeod and McCarthy. Now we know that it has to be DNA. This was the unequivocal proof that was needed, that it is DNA and not protein, and therefore the focus shifted on to the structure of the DNA. Now how to explain the diversity with the model of the DNA, and therefore people started working on the structure of the DNA, and this led to the development of the double helix model. And now we know that DNA can be very diverse. It can actually explain the diversity in the living world as well. So this question now is finally settled, that whether it is DNA or protein, and we say that